Hello and welcome. This is the second episode of the Introduction to Biotechnology course, titled Microbial Biotechnology. In this course and in this episode, you don't need any prior experience or knowledge. I am assuming your knowledge of biotechnology is zero. We will take things step by step to benefit from the course, follow the order of episodes. Now we have specialized. The first episode was an intro to biotechnology. Now we discuss each type. Today is microbial biotechnology. What should you focus on? Do not worry about side info, it will be explained later. Just follow this episode. First, what is a microorganism? Then its traditional applications? Then modern ones. A microorganism is a living organism. We agreed in episode 1 that biotechnology uses living organisms or parts or their products. No life, no biotechnology. Microbial means microorganisms. Most are microscopic bacteria. Algae, fungi, viruses are examples. There is debate about viruses as living forms. Many say they are not because they don't grow or feed outside cells. They only reproduce inside and have genetic material. So you can consider them in applications. Now to applications of microbial biotechnology. We briefly mentioned traditional applications before. Now, more details. First example is yeast in baking. Yeast gives taste and makes dough rise. Without it, bread looks and tastes bad. How does yeast make dough rise? By fermentation. It produces carbon dioxide gas, which inflates the dough. Alcohol is also made, but it evaporates in baking. Fermentation is a process where microorganisms get energy from sugars, converting them into alcohol, and gas fermentation is also used in making cheese and yogurt. In yogurt, bacteria give taste and texture. You can use a commercial yogurt as a starter, which contains the bacteria. In cheese, bacteria are used in making special types like Swiss cheese. The holes are signs of quality and made by gas bubbles. If absent, it is called blind cheese. During fermentation, gas expands like a balloon and makes holes in cheese. Another historical use is acetone production. During World War, acetone was made by fermentation. It was used in weapons. Now modern applications. First, penicillin production. The penicillium fungus produces penicillin. Humans provide a good environment. A medium with sugar. This increases fungus and its product. Then penicillin is separated, purified, and sold as medicine. Then recombinant proteins like insulin growth, hormone, and interferon. This is the base of genetic engineering. Recombinant DNA is DNA from more than one source. For insulin, we take human DNA. We cut the gene that makes insulin. We go to bacteria. Bacteria have plasmids, small circular DNA. We cut the plasmid. We insert the human gene into it, making recombinant DNA. We put this into bacteria. Bacteria multiply and express the gene. They make human insulin. We extract, purify, and pack it. So the insulin comes from a human gene produced by bacteria. It is human insulin, not bacterial. Same method for growth hormone and interferon. Interferon is used in treating viruses. Write a question or just show you watched. See you in the next episode. Goodbye.